Hello everybody. Today I will conclude with chapter 22. I will focus on nucleotide metabolism. I will begin with biosynthesis of purines and pyrimidines, and then I will finish with degradation of purines and pyrimidines, and also how nucleotide biosynthesis represents a great target for development of various medicines. Okay, so I will begin with nucleotide biosynthesis, and of course, before we start talking about how do we make nucleotides, it is good to revisit what is the role of the nucleotides. And usually we connect nucleotides as building blocks of nucleic acids such as DNA and RNA. And then we also connect them with chemical energy, but beyond that, we often do not really recognize the role of nucleotides in the function of the cell and in metabolism in general. So indeed they are building blocks for nucleic acids, and they are the chemical energy of the cell. But also they're involved as cofactors and also in the electron transport. NAD, FAD, CoA, these are all various forms of nucleotides that are part of the metabolic process, throughout metabolism in general. They are also involved in molecular activation, so for phosphorylation or when we do adenylation. And lastly, but not the least, they are signaling molecules, cyclic AMP and cyclic GMP. If you remember from 481 when we talked about biosignaling, they're major biosignaling molecule in the G protein coupled receptor signaling pathway. So nucleotides are not just building blocks for DNA and RNA. They're a lot more. Without them, basically life cannot exist. Without any of the biological molecules, life cannot exist, but ATP is the energy currency in the cell. Signaling, metabolism, enzymology, everything involves nucleotides. Okay, so similarly to amino acids, nucleotides can be synthesized either de novo or they can be recycled. So when we talk about recycling, that usually means that nucleotides are reused, or the bases of degraded nucleotides are reused for synthesis of new nucleotides. Our focus, though, today will be primarily on the de novo synthesis. And it's fairly complex, but again, I will try to make it as manageable as I can. An important thing that we need to focus on is to try to understand what are nucleotides are made of, what are the precursors. And there are four major precursors: amino acids, ribose 5-phosphate, carbon dioxide, and ammonium. From the amino acids, not all amino acids are involved in nucleotide production, but only a few: glycine, aspartate, glutamine. These three amino acids are primarily the ones that contribute for nucleotide synthesis. Glycine contributes for the carbon skeleton of purines, aspartate for pyrimidines, and then as sources for amino groups: glutamine for purines and pyrimidines, and aspartate and glycine for purines. Now, if you can't keep them straight right now, that's okay. When we start talking about the individual groups, I will highlight them again. But just amino acids: glycine, aspartate, glutamine. These are the amino acids that are involved in the nucleotide biosynthesis. Again, ribose 5-phosphate, but not in that form. The ribose 5-phosphate is actually activated by an addition of a pyrophosphate group at carbon 1. And then I'm asking you the question here: is this molecule prepared for consequent reactions? Why and what kind? whenever I look at this molecule, it reminds me a little bit of the activated isoprenes in the cholesterol synthesis, where we have 3 phosphates, one pyrophosphate group and then another phosphate sticking on the other end. And again, why were this pyrophosphate group there? It's an excellent leaving group. It's an activating group. By adding it to a molecule, even if it's stable, it destabilizes it and drives a reaction forward, usually a condensation reaction. So something will be added here on the first carbon. That's why this pyrophosphate is added to carbon 1. Carbon dioxide is a carbon source for purines and pyrimidines, and ammonia is a nitrogen source for pyrimidines. All right, so let's focus now on the biosynthesis of purines. As you can see from this schematic, it's fairly complex. We have a lot of building blocks pulled from various sources. However, in general, obviously, we're going to need a ribose 5-phosphate, or PRPP, 5-phosphoribosyl 1 pyrophosphate. That is the pentose, the activated pentose. And then, in terms of amino acids that are contributing for the purine synthesis, we have glycine and glutamine and aspartate as sources of amino groups, and as sources of carbon, glycine. So you can see glycine contributes amino groups and carbon, aspartate amino group, and then glutamine, sorry if I said glutamate, but glutamine contributes nitrogens as well. Additional carbon sources, carbon dioxide and N-formyl tetrahydrofolate. You can see that a tetrahydrofolate, well, it says here formate, contributes a couple of carbons as well. All right, so some of you might ask, oh my goodness, do I remember which carbon is contributed by who or by what? I would say keep in mind what are the contributing precursors for purine synthesis, and try to keep as straight as you can which carbon and nitrogen is being contributed by what. All right, so let's look at the specific protocol now. Something that is very specific for purine the pathway. Something that is very specific for the purine biosynthesis pathway is that the purine rings are synthesized while they are attached to the ribose. With pyrimidines, you will see that will not be the case. But purines, that is something specific for them. They are built while attached to the pentose. Okay, so the pathway starts with PRPP, and then, and no surprise, we have a substitution of the pyrophosphate by an amino group, and then glycine is added to that amino group with the help of ATP, and then we add another carbon to the amino group of glycine, and then glutamine donates another amino group and with the help of ATP, and again with the help of another molecule of ATP again, the 5-membered ring of the purine ring structure is now closed. It's still attached, as you can see right here, to the ribose. Then the pathway continues by addition of a carbon from bicarbonate that first gets added to the amino group and then gets transferred to the alpha carbon of the added glycine. Later on, we have an addition of another amino group from aspartate, and fumarate is released, and then N-formyl tetrahydrofolate donates one more carbon, and then the 6-membered ring structure of the purines is closed at that point. The base here is none of the bases yet that we normally recognize as purines. This is inosine, inosine monophosphate to be precise. And you can see how complex the synthesis of inosine is. And so try your best, but remember principles rather than specific names and anything in detail. One thing though I want you to realize, look, we used 1, 2, 3, 4, 5 molecules of ATP, and if we count also the ATP that was involved in the activation of the pentose, that's a lot of ATP was involved in the synthesis of nucleotides. All right, so the final product of purine of this pathway is inosine monophosphate. So from this point on, we will use now inosine monophosphate, and we will generate adenosine and guanosine monophosphate. Something that I wanted to point out is that the first step is the regulatory step of this reaction, and you will see later on when we talked about regulation, and this will be the step that will be regulated. Does it make sense? Of course it does. I mean, this is the beginning of the pathway. You don't want to regulate the last step. You want to regulate the first step. The same is with these pathways where we use inosine to make AMP and GMP. The first step of the deciding moment here, whether it's going to go towards G or A, is the regulatory step. So with the help of hydration and oxidation, with the addition, we basically add an amino group to the inosine ring structure, and we form guanine base. In terms of adenine, again with the help of aspartate and GTP in this case, we replace the oxygen with an amino group to form an adenine base. In terms of regulation, the final products of these reactions, of course, will regulate these pathways. You can see AMP, GMP, and IMP are all regulatory or feedback regulators of this pathway. You can see an example of concerted regulation where multiple products of these pathways regulate the same reaction. You also see an example of stepwise regulation where GMP, for example, can regulate, or AMP for that matter, can regulate the, for lack of a better expression, I can say the branching of IMP, deciding whether IMP is going to go towards adenine or guanine. But they also can regulate the first step of the pathway where PRPP is actually aminated with the help of glutamine. In addition to that, all of these products can, well, actually not all of it, it appears only at AMP. It regulates also the conversion of ribose 5-phosphate to PRPP. So you can see concerted regulation, stepwise sequential regulation. I don't know whether there is enzyme multiplicity here, but regardless, this is the regulation of purine biosynthesis. Now let's look at the pyrimidine biosynthesis. Now, as I've mentioned here, the difference with the difference of the pyrimidine biosynthesis from purine biosynthesis is that the ring structure of the pyrimidine bases is synthesized first, and then it's attached to the ribose. So again, this is something that is distinguishing between the two pathways. Now, what are the substrates for pyrimidine biosynthesis? Amino acids, aspartate and glutamine. Carbamyl phosphate, something that is familiar to you, but we'll talk about it in a little bit. Carbamyl phosphate will donate a nitrogen and a carbon. Carbamyl phosphate will be generated from bicarbonate or carbon dioxide and ammonia. The only difference with this carbamyl phosphate, though, is that it's going to be made in the cytosol by an enzyme, carbamyl phosphate synthase 2. you've heard about carbamyl phosphate and carbamyl phosphate synthase during the urea cycle pathway, but that enzyme works inside the mitochondria and again uses bicarbonate and ammonia to generate carbamyl phosphate that is used in the mitochondria for the urea cycle. This carbamyl phosphate, made again of carbon dioxide and ammonia, is made by carbamyl phosphate synthase 2, but in the cytosol of the cell, not in the mitochondria. Okay, so amino acids, aspartate and glutamine, carbamyl phosphate, which includes carbon dioxide and ammonia, and of course ribose 5-phosphate in the form of PRPP. So how does the synthesis actually happen? We start with an aspartate. Aspartate is fused with carbamyl phosphate to form carbamyl aspartate. Then the compound is dehydrated to close the ring structure, and it's oxidized to form orotate. Now this pyrimidine base now is attached to PRPP or to the pentose. The pyrophosphate will be removed, and orotate will be added to the pentose, forming orotate monophosphate or orotatidylate. And then when that molecule is now decarboxylated, this carboxyl group will be removed. We form the first common or known to us nucleotide, and that is UMP, uracil monophosphate. Uracil monophosphate then is phosphorylated to form uridine 5-triphosphate or UTP. Then glutamine, so we made UTP. So glutamine then is used with the help of ATP to exchange this oxygen with an amino group, and now we formed CTP. So how many pyrimidine bases are there? There's 3. And what type of nucleotide biosynthesis we've described so far? The ribonucleotides. So these nucleotides can only be used for ribonucleic acids or RNA synthesis. And of course, we've described the biosynthesis of A and G, adenine and guanine bases, and now we've described the synthesis of uracil and cytosine bases. The one base we haven't described yet is thymine, but that base only exists in deoxyribonucleotides. So in order for us to generate the thymine bases, we need to convert the ribonucleotides to deoxyribonucleotides. And that happens by following this fairly complex process, which we're not going to look into details. But what we need to understand from this is that ribonucleotides are precursors for deoxyribonucleotides. And you can follow the electron flow here in the chart, and you can see how the hydroxyl group in the 2' carbon is first protonated. How is this going to happen? To be honest, I have no idea, but the active site obviously promotes it. And then once it's protonated, water is now released, and then the resulting positive charge will be used to bind a proton from the environment. And then that's how we rip apart, basically, or rip out a hydroxyl group and replace it with a hydrogen group. Anyway, I've told you before that sometimes these reactions seem more, and that's probably the ignorant person in me that is speaking like that, but it seemed a little bit that we are trying to fit an explanation to actually what happens, because this is what we think it really happens. But like I said, that's most likely the ignorant person in me speaking this way. The interesting thing, though, to also understand is that this is one of the rare times where you're going to see radicals involved in the enzymatic process. and that's something, like I said, you would rarely see as an example in metabolism in general. Okay, so we have we generated the purine and the common purine bases. We converted all the ribonucleotides in deoxyribonucleotides. Obviously, we're not going to have, we don't have yet the thymine nucleotides, but this will tell you how do we generate thymidine monophosphate. And we're going to start with, we can start either with cytosine or uracil. They eventually, both of them will be converted after they're made into deoxyribonucleotides to DUTP. Then DUTP will be converted to DUMP, and then DUMP will be used as a substrate for thymidylate synthase that will convert deoxy UMP to deoxy TMP. Now this process has to be very tightly regulated, because if for some reason this process, there's something that interferes with this process, then you will have an increased concentration of DUMP or uracil, deoxyuracil nucleotides. And what's wrong with having deoxyuracil nucleotides? Well, uracil is not present in DNA. But if you have an increased concentration of DUMPs or DUTPs, they will be incorporated in DNA, and that would lead to various issues in the cell or in the tissues, such as heart dysfunction, cancer, brain abnormalities, which are all symptoms of folic acid deficiency. Someone would say, what does folic acid have to do with this? Well, folic acid actually is involved in the thymidylate synthase process of converting DUMP into DTMP. Thymidylate synthase uses methylene tetrahydrofolate, which is a derivative of all the tetrahydrofolates, I guess, a derivative of folic acid. But methylene tetrahydrofolate is converted to dihydrofolate. But then dihydrofolate reductase converts it to tetrahydrofolate, which then is converted to methylene tetrahydrofolate, which is absolutely necessary for thymidylate synthase to convert DUMP into DTMP. So if something interferes with any of these enzymes, then DTMP will not be produced properly. So I would say, well, it's a big deal. Well, actually, it is a big deal. I'm sure nobody would say that. But the problem also is that the DUMP will be increased, and then that will be incorporated into DNA that will cause all kinds of issues with DNA stability and structure. Okay, so we've covered how do we synthesize all nucleotides for DNA and for RNA molecules. Now, let's talk about about the degradation of purines and then later on of pyrimidines. So purines are usually excreted as uric acid or allantoin, or in some cases urea, but don't confuse urea with the degradation of purines. The most common degradation product of purines is uric acid and allantoin. So in terms of the degradation, we follow the following major steps. Starting from GMP or AMP, the first thing that happens to these nucleotides is the inorganic phosphate is removed. Then the base is separated from the ribose. Now these bases in the ribose could be recycled if needed, but if they need to be further processed and disposed of, then the bases, whether it's going to be guanine or inosine in this case, because AMP is converted to inosine during this step. They're all converted to xanthine, and then xanthine gets converted by xanthine oxidase to uric acid. Uric acid can further be processed, as you can see right here, to allantoin that most of animals used as a way of discarding purine bases. So this is how purines are degraded. Purines are basically released from the body or excreted from the body either as uric acid or allantoin. Pyrimidines, though, they undergo slightly different processing, and the major products of pyrimidine degradation are ammonia and carbon compounds that eventually get converted to succinyl-CoA and they enter the citric acid cycle. So as you can see, there are strict differences between pyrimidines and purines. Purines, again, uric acid and allantoin. Pyrimidines, ammonia that gets converted to urea, and then the carbon skeletons get converted to succinyl-CoA that enters the TCA cycle. All right, so nucleotide biosynthesis is a very lucrative or tempting pathway to be interfered with. And that is because, I mean, think about it. If you somehow stop the de novo synthesis or the synthesis of nucleotides, fast-growing cells will run out of building blocks. Now, most cells don't need newly synthesized or de novo synthesized nucleotides. Why? Because they already have their genetic material. They don't divide, so they don't have to multiply. They don't have to double it. They just use the genetic information. But fast-dividing cells constantly need to divide. And when I'm saying fast-dividing cells, these are stem cells and all cells in the body. I'm talking specifically in the human body or mammal's body that constantly need to be regenerated. Well, what are the cells constantly divide or have a very rapid division cycle? Cancer cells. What are the cells? Bacteria. So naturally, if we can control the conversion or the de novo synthesis of nucleotides, we will affect these fast-dividing cells because they're going to be the ones needing a lot of nucleotides. And in those cells, nucleotide synthesis will be active. So if we can somehow stop it, we can hold the progression of these cells. We can hold the division of cancer cells, or we can hold the division of bacterial cells. And so that's why many antibiotics and chemotherapeutics are actually used to block various steps of nucleotide biosynthesis. One of these chemotherapeutics is 5-FU or 5-fluorouracil. And this interesting chemotherapeutic, actually I've used it before in my research when we were doing transplant, but so what it does, it doesn't really exert its function directly. Instead, it gets converted to fluorodeoxyuracil monophosphate or fluoro-DUMP, which binds and inactivates thymidylate synthase. It basically, instead of DUMP, now we have 5-fluoro-DUMP that binds with thymidylate synthase and inhibits it. And that's the benefit. Then when we stop thymidylate synthase, then we're not going to have enough DTMPs. We're going to have more DUMPs, which, like I said, will start incorporating into DNA. DNA will not be able to synthesize because we don't have deoxy-T nucleotides. And so the whole process of division, whether it's bacterial cells or whether it's cancer cells, will be inhibited. Other examples are methotrexate, aminopterin, and trimethoprim. These are all various medicinal agents that inhibit now the pathway that regenerates methylene tetrahydrofolate from dihydrofolate. And specifically, for example, trimethoprim binds the bacterial dihydrofolate reductase on 100,000 times better than the mammalian. And this is important because you cannot just generate inhibitors for enzymes that are naturally also occurring in the human body. If you do that, then we will destroy or halt the division not just in bacterial cells but also the human cells. That's why actually chemotherapeutics are so toxic, because their function as inhibitors of cancer development is because cancer cells are actually a product of normal human cells or normal cells in general, whether it's an animal cell or a human cell, it doesn't really matter. But it's very difficult to control the division of these cells without controlling the normal enzymes that exist in the cells in our body that need to be regenerated all the time, such as the hair follicles, such as the lining of the gastrointestinal tract, such as the bone marrow, such as the reproductive system. And so that's why chemotherapeutics are very toxic, and usually when people take chemotherapy, in some cases their hair thins out or may fall off. They often develop diarrhea and vomiting because of that shedding and lack of regeneration of the epithelium of the gastrointestinal tract. Many of them develop anemia or other immunological issues, and oftentimes it leads to basically interference with their fertility, and many of them cannot have children. Again, this all depends on the type of chemotherapeutics. Some are more harmful than others, but in general, it's very difficult to use chemotherapeutics without affecting the division and the process of the normal fast dividing cells in our body. Another medicinal agent that is designed to block the degradation actually of purines is allopurinol. And allopurinol, as you can see, mimics very closely hypoxanthine. Look, the only difference is just swapping of these atoms. It's a very small difference, but with major consequences, because when allopurinol that mimics hypoxanthine and then it binds xanthine oxidase, it becomes oxopurinol. And once it becomes oxopurinol, the binding now of oxopurinol is much tighter to xanthine oxidase, and it locks it basically. The binding is so strong that xanthine and hypoxanthine cannot compete with oxopurinol to take it out of the enzyme, and as a result, we decrease production of uric acid. And most of the waste products of purine biosynthesis will remain as hypoxanthine or xanthine, which are just other compounds that can be evicted with urine. Now, some would say, well, why do we want to stop uric acid production? Well, this is especially helpful to people that have gout, for example. Or in this case, you can also see that acyclovir is another one of those nucleotide analogs that is used as an antiviral medication. But allopurinol specifically is designed to stop the production of uric acid, which uric acid, people with gout have a problem with the processing of uric acid. Uric acid in higher concentrations tend to crystallize, especially around joints. So people with gout, they have the typical pain in their big toe or around other joints, especially on the feet, but also in the hands, and it's incredibly painful because the crystals of uric acids irritate the capsule and of the joint, the ligaments, and it's incredibly painful. And so some people connect that with eating a lot of meat. Now, it's never been shown that actually eating a lot of meat starts gout or causes gout, but people that have gout are usually placed on a low meat diet because, if you can imagine, meat also contains, it's muscles, so they contain nucleotides, they contain nucleic acids, so obviously we're going to have an increase of uric acid production, and as a result, the increased uric acid would lead to more crystal formation and more complications with gout. So that's why allopurinol is designed to stop that, or to inhibit it at least, maybe not stop it, but to inhibit it, so you can give a little relief to the patients with people with gout, and to make life a little bit more bearable. Well, that's all I wanted to share about this chapter. I hope this was fairly clear. As you can see, I did not focus extremely on the details of the various pathways. I want you to understand them, but I don't want you to get bogged down in details. Yeah, that's basically in general all I wanted to say. Let me know if you have any questions. If not, I hope everything is well, and until our next time.